Hello, Taras here. Welcome to another episode of Ocean State Television. And today we are going to be talking about the other parts of the nitrogen cycle. Some of the more elusive biological aspects of the nitrogen cycle that aren't usually commonly discussed by aquarists, but I consider them extremely important, and frankly, in the future, they really are going to be some of the things that we start to use when we talk about the real difference between good tanks and bad tanks. Now, since we've been keeping aquariums, since people have been keeping koi ponds, since people have been rearing oysters, there have always been good ponds and bad ponds, good tanks and bad tanks. It's been an elusive concept, a concept where even the most skilled brewers of the day, at the end of the day, they didn't understand the difference between a good culture and a bad culture. They had no way of understanding the different biochemical mechanisms, just like we can't truly understand them today. But they knew that once they had something good, they could replicate it. And when they could replicate it, they gained power because they were selecting, they were acquiring that better yeast, that better critter. And today, that's why I think it's very important that we talk about other parts of the nitrogen cycle and some of the critters behind that. Before, we've talked about the conventional nitrogen cycle that guides all aquariums. So that's less so the difference between a good tank and a bad tank, but the difference between a tank that's all gonna die within 48 hours and a tank that's going to survive and then have a chance to develop towards greater complexity over time. So that's the conventional nitrogen cycle, where we have ammonia being oxidized and it's being converted into nitrite, and then further oxidized and it's being converted into nitrate by a different set of bacteria. We've gone through that. And we've also gone over denitrification, which occurs in the deeper layers of sediment in the anaerobic environment, where we have nitrate, that final end product, being converted into benign nitrogen gas and then escaping into the environment. Now today, we're going to be talking about the third and fourth aspects of the nitrogen cycle, which are far less commonly discussed in the aquarium hobby, but will be so more over time because they apply to everything. And they're also extremely important to gardening and the keeping of aquatic plants and terrestrial plants as we begin to have vivariums, paludariums, and keep boosting life above the rim, so to speak. Let's talk first about some of these plants here, particularly a freshwater mangrove and some of these other, like Tillandsia up here, the air plants. How are they getting their nutrients? It almost baffles. They live without soil. They're up there in the trees. Everything we know about plants is that they need soil to get their phosphates and their nitrates. So why is the Tillandsia able to live up in the tree seemingly without, any real soil to live on? Because plants, just like corals, we always treat a coral not so much just like a brute animal, but a careful complex of all of its different parts and composites and bacteria and microbes and fungi and archaea. And just like that, we need to treat plants the same way, because plants have this thing called the rhizosphere. It's basically all these different microhabitats on the plant that house all these different bacteria. And one of these bacteria, a class of them, are called Diazotrophs. And they're able to fix nitrogen gas. Now, what does that mean? It means that all that nitrogen gas over here is not available to plants. They can only use it when it's nitrate. Usually in the aquarium trade, we have too much nitrate, so we use plants to take it out. But if a plant is deficient, has no nitrate, there's a set of bacteria that are able to seize that nitrogen gas and form it into the nitrate that the plant can use. And that's an incredible power when we think about it. Because even though in the aquarium trade, we're always thinking in terms of controlling excess, we're thinking about how can we keep nitrates as close to zero as possible, because we're used to making those early mistakes of overfeeding, overstocking. But what about the situation where let's say we're at a coral farm and we have no nitrates and it's restricting our growth? How do we develop new and novel ways to introduce nitrates back into the system? If we need a system that can't be overfed with fuel, but needs a constant dosing of fuel, how do we get that perfect trickle, that perfect efficiency? And that is the key to the microbes. The microbes, the diazotrophs, they are the ones that are capable of being able to take that inseizable nitrogen gas and turn it into the fuel that plants can use to create all the different proteins, carbs, fats, and lipids, everything else. So the reason I'm going off on this is not only because embracing diazotrophs is going to be a whole other wave of being able to achieve higher levels of coral growth and get better symbionts for our plants, but it's also a good way to truly control the flow of nitrogen in the tank, which assists in making sure the fuel, so to speak, goes into the plants that you want, not the pest algae and the muck that you don't. Furthermore, it's also the key to sustainability in our world at large, because the way we make nitrogen fertilizer right now is by making ammonia through the Haber process, which makes fossil fuels, which we don't have a lot of right now. Last time I checked, it's almost $6 a gallon. So by embracing these critters that live in our tanks, we are also discovering new and novel ways to produce truly sustainable fertilizers, not only for our aquaria, but our greater world at large. Because at the end of the day, understanding this cycle is paramount to every single thing we try and do here. So that's diazotrophy. Now we're gonna be talking about the fourth part, and that's anammox. So first off, we were just converting ammonia into nitrite, then converting that nitrite into nitrate. That nitrate was then getting turned into benign nitrogen gas through denitrification, and then through diazotrophy, we learned how the nitrogen gas can be reformed back into nitrate. For most aquarists, we are mostly concerned with how to get rid of those base nitrogenous substances. Those are the ones that are most confrontational, the ammonia and the nitrite. Those are the things that can kill our aquarium animals, even in low concentrations. So, next process is one very recently discovered by science. It's very elusive, and even though it's highly hypothesized that it can occur in fresh water, to date we only really have evidence for it occurring in saltwater organisms, particularly deep water saltwater organisms in a varying mix of anaerobic and aerobic environments. So today we're going to be talking about anammox as well. So anammox is the direct conversion of ammonia into nitrogen gas. It's a way of basically bypassing the whole conventional system. And what makes it so exciting is that scientists believe that by learning from some of the deep water, saltwater microbes, and even the microbes that might exist in coral reef tanks and in the inner parts of the rockwork, especially those good tanks versus those bad tanks, that we can discover even more efficient ways of taking ammonia directly from a source, like let's say this cleaner shrimp, and immediately erasing it from the system. And by being able to do that, for our coral reefs, that's amazing, because we can strip nitrates like that if we want to, if we were able to harness this bacteria. But think about wastewater management. If we're able to strip nitrates directly from ammonia, the way we would be able to process wastewater in the future could be maximized. And this is another fantastic way where pursuing beauty in the aquarium also mirrors the sustainability that we have to seek out in a future and better world. That's a little bit into third and fourth parts of the nitrogen cycle. So every critter has ammonia when it eats anything. It excretes it when it burns those amino acids, and then ammonia can threaten our tanks. There's a variety of different ways that can get sucked up. But it's important to realize that there isn't one set of microbes doing this. There isn't one set of good probiotics or biofilter organisms. Nature is derived and stabilized, and the beauty we see around us is the product of all this infinite complexity. So in reality, we can't ever hope to just engineer our way into that complexity. We just have to find a way to live with it, foster it, and the aquarium is a good metaphor for that, because you're trying to achieve life and beauty at the same time. I'll see you next time.